位先生、各位女士，欢迎来到台湾免疫暨肿瘤学会主办的线上直播会议第2天 Post-SITC Conference 2020。首先，依旧让我们欢迎学会理事长张文震张理事长为我们进行今天的开场致辞。
各位电脑桌前的观众朋友大家早欢迎再回来我们今年的 Post-SITC 的 Conference。那延续昨天的一些成果，我相信今天大家还是会收获满满。那今天再跟大家提醒一下，我们2天的会后有抽奖活动。那这个抽奖活动，大家务必上去登录一下 QR Code， 那踊跃发问问题，那就有抽奖。我们有精美的我们 TSITC 的纪念行动电源供大家抽奖。那这是我们希望大家能够踊跃发言能够让大家在这个领域里面，让我们也能够办得更好。那今天很高兴那接下来我们会开始一个非常特殊的 topic， 是 Bioengineering 的 topic。那我们会欢迎我们赖志远教授他人现在在美国他会跟我们网络连线。那赖教授呢他是当时我是跟徐祖安理事在徐祖安理事的介绍下，我认识的赖教授他是 University of Washington Bioengineering 的 Associate Professor。那他当时我们聊到了，他说他可以用 Exosome， 然后因为 Exosome 是 Antigen 非常重要 Immunotherapy 非常重要的来源，他可以有一些方法，那可以来找到这些 Exosome， 然后让 improve 我们的 Immunotherapy。那我觉得这个非常好这个是我们非常非常需要了解跟学习的部分。那因此我就邀请他来跟我们指导。那他也很高兴，那也很乐意来跟我们分享那这边是非常感动，非常感谢他是这个没有 honorarium 来跟我们做这个指导了那我们知道了我们这些 oncologist 其实很辛苦了，治疗癌症的医生。那别的科别可能可以毕业之后，那就是继续执行他的医疗业务。那 oncology 的科别毕业才是开始我们需要了解非常多的 knowledge。我们还分身的部分，连基因的部分，那现在免疫的部分，让任何有新的东西，我们都要再次学习。那现在呢，我们昨天的会里面也学到，我们需要有新的一些科技，那新的了解，然后才能够追踪到这些细胞跑到哪里去。那今天也一样，那 Bioengineering 现在已经进来了。那我们呢，需要去如何找到这些 single cell。或者这些 cell 的一些 antigen， 那我们要如何去找到它，去 enrich 它，然后来 improve 我们 immunotherapy 的部分。那这个部分呢，我想今天赖教授的演讲会给大家非常大的启示。那在这边也呼吁如果还没有自己 career 的 young oncologist 这一个领域，也是大家可以去追求的。那我们现在就热烈掌声来欢迎我们的赖教授。赖教授，请。
Thank you, John, for the kind introduction. Thank you for the invitation. So my name is James Lai, and as John has already introduced me, I'm a research associate professor in the Department of Bioengineering at University of Washington. And then, so before we start the research discussion, I would like to just do a little bit more infomercial. The photo that I show on the background of my slide, this is a place we call Mount Rainier, and then it's a great place to do bike ride during the summertime. Winter would be too cold for that. And then so hopefully after the pandemic, we will be able to have you come over here, and then we can enjoy the sightseeing together. And then so today what I would like to do is to share some of my research activity related to the immunotherapy. And I'm doing this slightly different. My background is trained as a chemical engineer. So I've been looking at the field in terms of what are the challenges that the engineer can help to provide some solutions. Okay, and then so hopefully this will give you a slightly different view in terms of immunotherapy. So the key role of the immune system in cancer has been recognized for decades, but only during the past 10 years, the immunotherapy has been established as a major cancer therapy. So, if we think about, let me see, actually, before I do that, I do want to mention one thing. The meeting that you have is called a Post-SITC, right? So I did go to the SITC 2019 last year, and they hosted at the same location every year as the National Harbor. And then this is when you look from outside what the convention center looks like. And then if you look at inside out, and this is what it looks like. And on top of that, this is actually one of a meeting that I feel probably the most intensive in terms of people who are really focused on learning. If you ever get an opportunity to try that, I highly encourage it. It is the meeting that definitely feel like worth every penny. And there's a lot of information. All the ongoing clinical trial, things like that would also be discussed in the place. And then so back to the discussion of a science, if we think about using the triple negative cancer as a model, this is a cancer that all tumors are lacking the expression of an estrogen receptor, the progesterone receptor, and the HER2, and which is about 15% of breast cancer patients. And in general, this particular cancer has a very aggressive clinical behavior, and it also lacking the recognized molecular target for therapy, and therefore the prognosis is relatively poor, right? And that being said, the chemotherapy remains still to be the primary treatment option. And then they also have more frequent reoccurrence. And the finding of the population of a triple negative breast cancer is immunogenic and also actively engaged by the immune system provides a very strong rationale for testing immunotherapies. And then I'm not going to really tell you all the clinical trial ongoing for the in terms of the information, because these you can find them pretty easily. But when you think about this during the past few years, especially since 2016, the two sort of a common immuno checkpoint inhibitor that we know, one is the pembrolizumab and the other one is atezolizumab. They're only showing somewhere about 20% of the overall response rate, which is relatively poor from our standpoint of view. And these are already considered the patient that has been pre-selected based on the overexpress of the PD-L1. And one of the things, interesting enough, point out in all these studies is really stress the analytical assay that are being used for the biomarker assessment are crucial for these trials, because we're relying on these assays to do the initial patient selection. We're also relying on the assays to do the evaluation during the treatment. Right? And then so the biomarker assay that we're currently using is still suffer from a very significant variability from the pre-analytical, analytical, and post-analytical. And so we've been seeing huge amount of a discrepancy from one clinic to another, from one lab to another. And these are the issues really needs to be addressed urgently, because all the trials are based on these data, to select patient, to know if the outcome is proper or not. And so, for example, immunohistochemistry is one of the most commonly used gold standard for the tumor tissue microenvironment characterization. And when you look at it, it's a simple thing. You put an antibody, treat the tissue. Based on the antibody recognition, you will know whether the tissue has the marker you want or not. However, the IHC really lacks sensitivity. It has a very significant variability for what we call the assay to assay, lab to lab. Right? And it's only at the best to be semi-quantitative. And then it's very hard to standardize it. With all the standards that we know, try to implement it, it's still really hard to say, well, for this lab, when you use this, it's the same as the other lab. And then let alone nowadays, all all the biomarker assessment require what we call the multiplexing, meaning you need to detect multiple biomarkers simultaneously. And that is also very limited by the IHC. And so to address the challenge, my colleague at the Fred Hutch Cancer Research Center, Mandy Pavlovich, they developed what we call the multiple reaction monitoring mass spectroscopy, meaning to use for precise tumor tissue biomarker assessment. And we call it the MRMS. Okay. This particular assay is complementary to immunohistochemistry. Here are the data generated from Mandy's group, is showing the correlation between the mass spec assay and the IHC. So on the bottom of the x-axis is showing the negative and positive characterized using IHC. On the y-axis is showing the concentration of a marker that the mass spec is capable to detect. And so you can see for the HER2, it has a great correlation with the IHC, and same thing for the estrogen receptor. In addition to the correlation to the IHC, they also investigate the correlation between the fixed tissue versus the fresh tissue. And this shows literally a very linear correlation. So we know the assay is highly robust, and it can be used for different specimens, and it can easily be used to multiplex for many different things. And they have already characterized the assay very extensively, including to carry out the assay evaluation at a multiple different sites, internationally, domestically. They also carry out a large-scale assay evaluation, and including they can put the radioisotope in there as a characterization, as an internal standard. Okay. So Mandy's group, in addition to develop the assay, they also work with the National Cancer Institute to develop a clinical proteomic tumor analysis consortium we call CPTAC. And CPTAC is a public repository of well-characterized mass spec-based target proteomics assay. The purpose of a CPTAC assay portal is to facilitate a widespread adoption of target mass spec assay by providing a standard operating procedure reagent as an assay characterization. So you can see here on the diagram for the CPTAC in terms of the pipeline, it goes from the original tissue source, and which would go to the biospecimen core resources where they have the pathology and the molecular QC for the specimen. And then the specimen would also be sent to the proteome characterization center and later got fed back into the database. And the data then would be analyzed by the proteogenomic data analysis. And then there's also a translation research center there. They're both taking the data to do further analysis, feed it back to the database. And then at the end, the research community would be able to query the question they're interested in knowing, the assay they would like to develop, also the reagent they needed to be available by doing the search in the database. Okay. And then, so these all works out really well. You know, the process that Mandy's group have to develop, starting from taking the tissue from the patient and then fix the tissue. After that, the tissue would be dissect or take a section of a tissue, melt the wax, and then lyse the cell. From there, they would go through some protein digestion. And then the last step would involve using the antibody magnetic particles to isolate the target peptide. And then before they will feed it to the mass spec for the detection. And this particular process works really well for her group as well as all the others, as I described previously. They've been doing all the evaluation. However, the challenge that they've been experiencing, and this is when Mandy and I have a discussion about it, as a bioengineer, we've been trying to see if there's something we can do together, is one of the things associated with the assay is it requires a significant amount of tissue. In another word, in our world, we call the limit of detection being a challenge. And so what happened is, in order to go what we call the signal above the noise in the mass spec, they need to take a huge amount of a tissue from the patient, which, you know, often enough is not literally available. And then on the other hand, often enough, you want to use what we consider to be the minimum invasive approach to get the tissue from the patient before you want to do any analysis. And then, so this is one of the major challenges that they've been experiencing. They often have to get enough of a tissue before they were able to do the biomarker assessment. And on top of that, the current process they have is 15 manual steps, and which takes about 38 hours or so. The time is probably not critical. If we consider the cancer patient, usually waiting for a day or two is not the end of the world. However, the 15 manual step is another thing that we are hoping to address, because the more user steps usually means more training for the technician, and it would also create additional challenges. We have experience on something similar. This is more for the HIV drug-resistant diagnosis. What we found out was when there's so many user steps, the assay can go left and right, up and down. You just cannot predict the outcome for those. So we're hoping using our technology, we can address the first 2 points. So when we heard the challenge, we look into it. And one of the things is actually not a new issue. It's really about the magnetic particle that they're using to isolate the peptide. And so this is a comparison that I'm showing you here. And it's a very standard conundrum that what we've been observing for the magnetic particle affinity reagent. Currently, the mainstream is using the magnetic microparticle. You can put an antibody on the surface to recognize the target you want. These are the particles typically more than a micron, or you call it 1,000 nanometer. It's very good for the magnetic separation. It goes super fast, takes about a minute or two at the most. Unfortunately, when you look at the particle size, do the analysis for the binding, what we are finding is their kinetic is very slow. Their efficiency is relatively low. Ideally, to improve or to address the challenge associated with the kinetic and the efficiency, you want to go for the magnetic nanoparticle, or just a small particle in general. You can make them the size to be similar to the protein. And then so you will be able to achieve a very fast recognition. You can also achieve a very high efficiency. However, on the other hand, what we're experiencing is when we make the particles so small, we cannot separate them in real time. It would typically take about 8 to 10 hours to do the separation. So in another word, you cannot have your cake and eat it. And that was the challenge when we started the project. And so we decided to broaden the polymer technology that we have. We've been using it for decades or so. This is what we call the smart polymer. It has a very sharp and large response to small stimulus. What I'm showing you in the video is a temperature responsive polymer. On the right-hand side, this is a warm water. On the left-hand side is ice water. So you can see the polymer solution originally was transparent. When it see the warm water, it will transition from transparent to become opaque. And that's because the polymer would transition from hydrophilic to hydrophobic. And then this particular behavior is reversible, meaning when you put the polymer back to the cold water, it will transition back to become transparent again. So we decided to put these polymers onto the surface of the magnetic nanoparticles that we have, because it will allow us to reversibly aggregate the particles. So when the particles are small, that will allow us to achieve a very high efficiency as well as a rapid kinetic. And then when we aggregate a particle together, then we can achieve a very rapid magnetic separation. And so we spend a lot of time on the synthesis of these, but I'm not going to share with you about the chemistry. All these has been published previously. But what I'm showing you here is a video of how we separate the magnetic particle in real time. On the left-hand side, this is a neodymium magnet, and now you can purchase them off the shelf. On the right-hand side, this is a magnetic particle that we synthesize in our group. And then so what you're going to see, instead of using the temperature to trigger the polymer transition, here we're using the sodium chloride. It changed the solution ionic strength. So in the beginning, you can see there was no magnetic separation because, as I described before, these particles are way too small to respond to the magnetic field in real time. And somebody just add a sodium chloride solution, which triggers the polymer transition to be hydrophobic. Once the particles are aggregated, and you can see the separation happen in real time. So this has been the core technology for many of our development, including the mass spec assay as well as the exosome project that I will share with you later. And then so using this, we did a first demonstration, tried to develop a different affinity separation. Traditionally, we've been attached the antibody to the magnetic particle because the separation of a particle would occur together with the antibody. However, because of the technology we have, we're able to separate the two reagents as a binary reagent system. Now the antibody is independent away from the magnetic particle. As long as they have the same polymer modified, then we can trigger the temperature or pH shift, and it will aggregate them together to facilitate the separation. This particular strategy from the engineering point of view has one significant advantage. It's because now we can add as much antibody as we want independently. That would drive the antibody antigen recognition to the right-hand side, meaning potentially we will get higher signal compared to the noise. And then so the first demonstration we did was to show Mandy's group, compared to the magnetic microbeads that her group has been using, what would be the difference? The first difference that we are able to see is the kinetic of it. So the x-axis is the time for the incubation of the reagent and antigen. The y-axis of the data here is showing the capture efficiency based on the ELISA that we did. So when we use the DyNAO-B, which is what her group has been using during the past 10 years or so, you can see the starting point of a capture efficiency was about 20% or so, and then slowly increased to 70% when we finished the comparison. And when we use our polymer antibody conjugate, we immediately started from the 80% and then reached a plateau, which was about 90% or so within a few minutes. And then so when we did the simulation to look at exactly how much time it would take for the microparticle to catch up with our polymer antibody conjugate, it looks like it would take about an hour or so. So for those of you, if you ever actually try the DyNAO-B reagent, you might find, interesting enough, they always describe the incubation at a 4 degrees Celsius for an hour or longer. And this is really where it comes from. We did this study ourselves to realize that. And then so that's one of the advantages of using our reagent. We can really speed up the processing time because our reagent doesn't require much of an incubation time compared to the magnetic microparticle that has been utilized. Another thing is we try to use this together with the current biomarker that Mandy's group are trying to identify in the tissue specimen. And this is, if I remember correct, this is a CEP-C that they're using. Is based on one peptide. I'm showing you here. There are multiple peptides that we try to analyze, but I only get you one data set to represent it. So you can see the original reagent that the Fred Hutch has been using has about a 20 unit here based on the relatively peak area. And then so when they use our reagent, which is the number 2 and number 5 here, you can see there's a significant increment from the 20 to the 25. Right? What this really suggests is using our reagent, we can capture the peptide more efficiently. We can potentially increase the signal from the original 100% to 120%. And that being said, this also shows the potential we might be able to, using this technology, to use a smaller amount of a tissue, which is really what the grand challenge that we're hoping to address. So we have an ongoing study as we're speaking that was started early next month. And then so hopefully, in the recent future, we were able to show you the data or new data that we're able to use this particular reagent technology to reduce the size of a tissue down to a potentially a needle biopsy. And that's really what we want to do. Another piece about using the needle biopsy tissue is about the apparatus. Currently, Mandy's group has been using mainly just a very excellent research scientist to pipette all the solution. But that's not entirely feasible for the small piece of a tissue, especially when the desire is not to dilute the tissue specimen too far. because it would impact capture efficiency. And then so what we're hoping using a different piece of a technology in our group to address the challenges, instead of doing all the manual pipetting, we're hoping we can implement the whole process into a microfluidic device. This is part of a device, and on the left-hand side, this is a CAD drawing that my student did, and then they designed the device into two pieces. The left yellow piece is for the reagent loading. The right gray piece, this is for the tissue processing and the peptide isolation. And then so we recently established a reagent loading. The way that we're trying to do is to preload all the reagent into the channel here. Between each reagent, there will be air gap to separate them. So these reagents won't be pre-mixed. And then from there, all we need is a single pump, and that will pump all the solution into where we want to digest the tissue to process the tissue in one shot. And that will streamline the process. We anticipate if we can implement the process onto the microfluid device that we designed here, we will be able to reduce the number one, the tissue specimen down to less than 5 milligrams, which is about the size of a needle biopsy. We can also reduce the original 38 hours process down to 8 hours or so. And it would also allow us to reduce the user effort, because the original thing was about 15 manual steps. And I think with this particular strategy, we might be able to reduce it down to just few manual steps. And this is the data that we are showing you here for the reagent loading. The reagent loading is relatively clean, and you can see the first reagent and the second reagent, they literally have no spec overlap. And then so from here, I will switch the gear a little bit to the exosome. So the exosomes is a type of extracellular vesicle containing protein, DNA, and RNA from the cell that secreted them, or we call them the parent cells. And these exosomes will be taken up by a distant cells where they can affect the cell function and behavior. Lovo, and others have shown the function of an exosome back in the 2007. Some cells are actually using exosome to transfer genetic materials, and it would help to regulate the expression of a gene. And then they use this particular strategy to communicate with other cells back and forth, and which is really the basis for a lot of exosome drug delivery project or drug delivery potentials. So here I'm showing you one of a company that are hoping to commercialize their exosome technology for therapeutic. The company is called Kodiak. Kodiak is a Boston-based biotech startup. They develop a technology called the Injex. The Injex technology allows them to engineer the exosome to have the surface receptor that they want, as well as the content inside of it. And this is a cartoon to represent that expression. And then what they're interested is using the exosome technology to activate stimulator of the interferon gene, or what you call it STING, to enable cancer immunotherapy through the induction of a variety of pro-inflammatory cytokine and chemokine, including the type 1 interferon. And so what Kodiak has found was if we would administer the STING agnostic into the tumor intratumorally, it would show the rapidly leak out of the tumor and then causing the systematic adverse event and direct immune cells ablation in the tumor microenvironment, which is really what we don't want. And then so what they've shown using their technology that they're able to package the STING agnostic inside of an exosome, and then they're also able to produce the exosome with PTGFR on the surface, which is a marker that they're hoping to use for more targeting effect. What they found in their study suggests when they administer the exosting, as they call, it will generate potent, target, systematic anti-tumor immunity without the inflammatory cytokine-driven adverse event. So which is a step forward. And originally, they're hoping to start the trial before June this year. I haven't followed up with that. So, with the pandemic, maybe they would have some delay, but hopefully they would start it soon. This is truly one of the exciting technologies that I'm hoping to see it comes through. The other one that associated with the exosome that we know is, other than using it as a delivery vehicle, the exosome can also be utilized as a biomarker because they carry the molecules from the parent cell. So whatever happened to the parent cell potentially reflected on the exosome content. So City of Hope, they initiated a clinical trial in 2016, aiming to combine the pembrolizumab as well as the enobosarm to treat a metastatic triple negative breast cancer patient who are overexpressed with the androgen, which is another hormone receptor. And then, so in addition to showing the combined effect might be able to treat the patient, what they're also hoping to explore is to evaluate the effect of the combined therapy on the tumor-derived exosome, they call TEX, and as well as the biomarker associated with the TEX. And this would, if this comes through, it can potentially be used as a companion assay, which will be used to evaluate the treatment outcome side by side when the patient is receiving it. So all these trials represent a potential exciting future. However, one thing that sort of often be, I want to say, neglected is thinking about the process to manufacture the exosome. Over the years, we've been seeing all exciting research how a particular biomarker or particular molecule can treat a patient. But often enough, by the time when we want to commercialize it, one of the biggest challenge we have is how do we produce them at a large quantity with a very high quality that would allows people to use it safely. Okay. And then so exosome is actually right on the horizon. Biologics like a monoclonal antibody went through this particular process already, and exosome is a new one. So currently the separation and this requires very sophisticated and as well as efficient separation of the exosome before you can actually consider a mass production. So currently the exosome isolation are limited by a few different methods, or commonly utilize few different methods. The first one is we call the ultracentrifugation. So you put a solution of exosome in there. In some cases, you can use sucrose or sugar to create a density gradient, and then you spin them at a super high speed. And then so the exosome, due to the density, they will be partitioned at a particular space in the solution, and that what allows you to do the effective separation. This particular approach takes time, which is not the deal breaker per se, and it can achieve the purity that you want. However, it has a very limited capacity. When you think about you need to have an instrument like this, and of course, for the mass production, the instrument is going to be much bigger, and then you need to spin them sort of a batch process. So that's not very ideal. However, this is the method that Kodiak has been using. They're doing the centrifugation with the gradient. Another method that in research, I don't know if in reality anybody is using it, is called the precipitation. This is really mixing a polymer solution with the biofluid that containing the exosome you want. And what happened is the polymer solution, because of the solubility, is going to occupy most of the water molecule, and then it will precipitate out the exosome that you want. So over time, you're going to see your exosome precipitated, and you can do a single or quick centrifugation to isolate them. And this is actually not a new technology. In the old days, when we're still doing the protein expression in the lab, this is how we precipitate it. The particular approach is very simple. It doesn't take much of a time per se. However, it doesn't give you the purity that you want. Okay, it's going to precipitate a lot of things that you don't want. And then another approach that people use is size exclusion chromatography. They're actually commercially available size exclusion chromatography that you can use. It will give you that pure exosome fraction, however, is not really going to be, it cannot be scale up at this point, and that's one of a challenge. And then another method people use is filtration. This is really just based on the membrane with different pore size, allowing the exosome to flow through. Sometimes they would have a tangential filtration as an upstream process to remove some of the small molecule that we don't want. And then again, this particular process doesn't give you enough of a purity and yield you want. And so all the four methods that I show you here, they are really based on the physical property of an exosome. They cannot really help you to access the biochemistry information, which is what we really needed. And so the only method that we know so far is using the magnetic microparticle to do the exosome separation based on targeting the receptor on the surface. This particular process, again, is not, you cannot really scale it up, and it's time consuming. And then so they're not the best fit for the mass production. However, we've been interested in this field for a long time, trying to help the study to move forward in a different fashion. And so what we've been doing is using our smart polymer reagent that I showed you previously to help to isolate exosomes from human plasma directly. We can also do this from urine, from cell culture media. We can scale this up to literally any size that can be that is needed. And so here's a demo that we did. We published a paper back in 2018. So we have some solution. These are the seminal EV, so they're extracellular vesicle from the semen that we collect from the male patient. And then we're using our antibody polymer conjugate and the magnetic particle to target what we call the tetraspinin. These are the common markers on the exosome. We're not targeting any specific disease marker yet. And then the downstream analysis we use to quantify our separation is using the RT-PCR to quantify the microRNA inside of it. And so you can see when we use the tetraspinin antibody, we capture a lot more microRNA that we targeted compared to our control groups here. And then so here's in summary, what I've shown you is our smart polymer reagents and then how we are using it to facilitate the simple preparation, and which can be used for the mass spec analysis to facilitate the tumor tissue microenvironment characterization. We're also interested in using that to facilitate what we call the biologics production. The other half of the group actually also working on using the same technology to improve assays like an immunoassay to make it more sensitive and more specific. And then so here's the acknowledgement for people who, including collaborator in Fred Hodge, Jake Kennedy, and Mandy Pavlovich. Lucia Wojtek and Hillary Gamel were the two that I collaborate on the exosome project. And then Pat Staten and Ada Hoffman, who has been my long-term mentor. The funding sources are here. And I do have one more message that I want to share with everyone. Since the pandemic, we've been locking in our house for a long time. And I only go to the office 2 days a week. And recently, my daughter was able to go to one of a nearby, we call the Golden Garden Beach. And this is the message that she wants to share with everyone, as Taiwan can help. Thank you.
Great. Thanks, James. Thanks for the inspiring talk. And we have a couple questions. I'm not sure if you can see it online.
How do I see that?
It's supposed on your slide. Supposed on your slide. If not, I will read it for you.
Okay. Got it.
Okay, and then you just go ahead and answer two of them. Thank you.
Sure. So can we use this system to analyze neoantigen? I think the answer should be yes. And the reason I'm being a little bit more conservative is because I'm trained as an engineer. So I can tell you the fundamental principle behind it is all we need is an antibody that will recognize the target that you want. Our system has been utilized in the blood, in urine, in saliva, in semen, independent of whatever the target that we're interested. So as long as there's an antibody that we can utilize to isolate neoantigen, shouldn't be a problem. The only reason I keep saying should is because we haven't tried it yet. And then the second question is, can we use the polymer as an antigen to pause immunocyte for cell therapy? That's an interesting thought. Unfortunately, I have to tell you that I don't know. I don't know. The polymer itself, I don't know. The reason why I keep saying I don't know is because the polymer has been used for some in vivo study. However, I'm not very familiar with their immunogenicity associated with that part, and that's why I'm a little bit less certain about it. But I would like to sort of give you a hand-wavy thing here. We traditionally is more a polymer chemistry group, and we have been able to engineer the polymer composition to enable different applications. So, you know, I think we can at least give it a try, but the specific idea would have to be discussed, and I'll be more than happy to talk about it offline if anybody is interested. And I thought I might have thought, okay, so here's another one. So the third one is, in the small polymer model, is it possible to differentiate exosome from other microvesicle or unbounded secreted protein aggregates? The answer, I think I would say yes. So, but still, this is meant to be tested. So our reagent system is really based on the recognition from the antibody. So whatever the antibody will be specific, that's what we're able to do. However, there's a subtlety between the microvesicle and the exosome. People talk about maybe the size being the different, but there are also discussion about maybe the biomarker are different because the way how they're being produced are different. One is just directly budded from the membrane. The other one actually goes from the endosome or the intracellular space. And so my understanding is we've been able to isolate the exosome from the microvesicle based on the biomarker displays it on the surface. And so that I know we can do it. For the protein aggregate, if it's the same protein that displays on the surface of exosome, our current strategy won't be able to differentiate it. This might require some sort of an upstream separation to remove the protein in advance.
Thank you, James. 我知道赖教授他都会讲中文啦，哈。那我也帮我们这边，因为学会这一次的会议系线上也有护理人员，也有药师人员。那我想帮他们再问一个比较common 的问题，那可能赖教授也麻烦用中文回答一下。也就是说，我们这个系统也好，或者是 Polymer 或者是 Exosomes 这一些，似乎可以当成一些新的 antigens 的来源。那我们也可以用到一些比较少量的 tissue， 然后可以取得很好的 antigens 的来源。那 antigen 是我们在 Immunotherapy 上面，那除了药物之外，现在我们台湾也有特管办法细胞疗法的部分。那不晓得利用这样子的 antigen 可不可以改善我们的免疫治疗以及细胞疗法？赖教授请。
细胞疗法的部分哦，这个我自己没有 involve。就是没有参与啊，但是我知道，因为我们自己有一个 spin off 的 company， 他们有在那个细胞疗法这一块有做过一段时间。那我们的那个参与其实是在上游的部分，就是拿我们的这个polymer 的 reagent 拿来做那个细胞的生产，就是把我们需要的这些免疫细胞先做分离。然后呢，可以透过这些 polymer 的 composition， 可以去针对细胞做 engineering， 像我们讲的那个 CAR T cell 会有一个 chimera receptor 这些东西，对我们的 spin-off company 之前有尝试过。那我的了解是说是可行，但是因为我跟公司之间有 conflict of interest， 所以这一块我比较没有直接参与。
好，谢谢赖教授。那希望说将来有这个机会，可以在台湾，我们可以一起合作。那学会的会员也好，或者是学会里面的各个成员也好，我们都可以跟赖教授再做学习。好，那我们再次热烈掌声，谢谢赖教授，谢谢。
谢谢，谢谢。
谢谢赖教授以及张理事长。